After studying this module, you shall be able to know the role of metal ions and other inorganic elements in biological systems. Learn about the role various metal ion plays like sodium, potassium, lithium, manganese, zinc and calcium. Learn about the various examples involving metal ions and their complexes playing important role in biological systems. Identify the importance of different enzymes in proper functioning of organisms. Inside the human body, there is a system helps in managing and regulating trace metals in key amounts and circulating in blood and stored inside the cells. From our diet, nutrient metals are incorporated into blood, transported into cells if the level of nutrient is depleted in cells. They also help in excreting out of nutrients if the amount is overloaded. If due to some reason this system stops functioning, abnormal levels and ratios of trace metals can develop inside cells and blood. Commonly, trace metals disturbing the level is elevated copper and depressed zinc. The fixed ratio of copper to zinc is very important, even than their individual concentrations. Inside the human body, amount of zinc present is approximately 2 to 4 grams. Mostly zinc is present inside the brain, muscle, bones, kidney and liver. But the maximum amount is inside the prostate and parts of the eyes. After iron, zinc is the second most abundant transition metal and it is present in almost all classes of enzymes. Copper is also a dietary nutrient. It requires in small amount. Copper is the third abundant trace metal. The total amount of copper inside the body is only 75 to 100 milligrams. It is in every tissue of the body but stored in the liver with fewer amounts found in the brain, heart, kidney and muscles. Metal ions in biological system. Sodium. Sodium is a major component of the cations and the extracellular fluid. It mainly exists as chloride and bicarbonate. It helps in regulating acid-base equilibrium. It also helps in the maintenance of the osmotic pressure of the body fluid and thereby protecting the body against exclusive fluid loss. Its role in the preservation of normal irritability of muscle and the permeability of cells cannot be ignored. Potassium. Potassium is present as cation in intracellular fluid as well as in extracellular. Within the cell, it functions just as sodium in extracellular fluid by influencing the acid-base equilibrium, osmotic pressure and water retention. It is essential for several important metabolic functions, including protein biosynthesis by ribosomes. When present in extracellular fluid, it influences muscle activity. In addition to the mentioned role of potassium ion, a number of enzymes such as glycolytic enzyme pyruvate kinase requires potassium ion for maximum activity. As mentioned, most animal cells have higher concentration of potassium ion inside the cell membrane and higher concentration of sodium ion outside the cell membrane. The sodium gradient hypothesis of glucose transport is linked to sodium potassium ion transport known as sodium potassium pump. Sodium potassium pump or sodium potassium exchange pump is shown in figure. As you can see, two potassium ions go inside the cell instead and the three sodium ions comes outside the cell, that is in the extracellular fluid with the help of sodium potassium exchange pump. ATP is required in this sodium exchange pump which is converted to ADP and PI, that is a phosphate group. Lithium. Lithium ion in form of lithium carbonate can be used in the treatment of manic depressive illness. Lithium ion is present in intracellular and extracellular fluids and has tendency to replace potassium ion as an activator of sodium potassium ATPase. Although ATPase activity is less than lithium ion, it is present then when potassium ion is present. Calcium. Calcium is present in the body in large amount than any other mineral element. About 99% of the body calcium is in the skeleton, where it is maintained as deposit of calcium phosphate in soft fibrous matrix. The very small amount of calcium not present in skeletal structure is in the body fluids, where it is the part ionized. Indeed, this small amount of ionized calcium in the body fluid is of great importance in blood coagulation, in maintaining the normal excitability of the heart, muscles and nerves, and in the differential aspects of membrane permeability. The matrix of bone in which calcium is deposited has a unique structure essential to normal calcification. The major inorganic constituent of bone is comprised of a crystalline form of calcium phosphate, closely resembling the mineral hydroxyapatite. In addition, however, the bone contains a substantial amount of non-crystalline amorphous calcium phosphate. It appears that this amorphous material is predominant in early life, but is superseded in later life by the crystalline apatite. Although it may appear that the mineral deposit in bone are more or less permanent, this material, like almost every other constituent of the tissue, is an actual fact in a dynamic state, constantly being formed and resorbed more rapidly during early development and at a slower declining rate during adult life. As shown in figure, this shows the regulation of calcium, how calcium is reabsorbed in bone, how calcium is absorbed in intestine, and when there is an increased amount of calcium in blood, it goes to parathyroid gland and parathyroid hormone. Manganese. Manganese is essential for reproduction, normal bone structure and functioning of the central nervous system. Zinc. Zinc plays an important role in cellular metabolism. Zinc is bound with 10% of human protein, which helps to transport zinc in required area. It is used in the catalytic activity of more than 200 enzymes and also plays an important role in immune function and increased protein synthesis, DNA synthesis and cell division. It supports normal growth and development during pregnancy, childhood and adolescence. It possesses antioxidant properties, protect against accelerated aging and accelerate the healing process after an injury. However, studies differ as to its effectiveness. Zinc ions are effective antimicrobial agents even at low concentrations. Gastroenteritis is strongly attenuated by ingestion of zinc, and this effect could be due to direct antimicrobial action of the zinc ion in the gastrointestinal tract, or to the absorption of the zinc and re-release from the immune cells. All granulocytes secrete zinc, or both. Cells in the salivary gland, prostate, immune system and intestine use zinc signaling as one way to communicate with other cells. Inside the brain, zinc is stored in specific synaptic vesicles by glutamatergic neurons and can control brain excitability. It plays a key role in synaptic flexibility and so in learning. It also can be a neurotoxin, suggesting zinc homeostasis helps in normal functioning of the brain and central nervous system. Zinc deficiency results in various disorders as it affects our immune system. It is shown in figure. Due to the deficiency of zinc, it affects the immune system. Zinc deficiency causes abnormal development of human organ. The thymic T cells do not function normally and these become susceptible to infectious increases. Copper. Copper plays an important role in our metabolism, largely because it allows many critical enzymes to function properly. It is important for strengthening skin, blood vessels, epithelial and connective tissues inside this body. Copper plays a role in the production of hemoglobin, myelin, melanin, and it also keeps thyroid gland functioning normally. Copper can act as both an antioxidant and a pro-oxidant. Free radicals occur naturally in the body and can damage cell walls, interact with genetic material, and contribute to the development of a number of health problems and diseases. As an antioxidant, copper scavenges or neutralizes free radicals and may reduce or help prevent some of the damage they cause. When copper acts as a pro-oxidant at times, it promotes free radical damage and may contribute to the development of Alzheimer's diseases. Maintaining the proper dietary balance of copper along with other minerals such as zinc and manganese is important. As you can see in figure, memory loss occurs because of the deficiency of copper. Examples of metal ions in biological system. Some more examples involving metal ions and their complexes playing important role are given. Carbonic anhydrous. It is a metalloenzyme containing 1 gram iron of zinc ion per peptide chain, which is essential for enzymatic activity of carbonic anhydrase. The structure of carbonic anhydrase, which shows the presence of zinc, is shown in figure. The biologically important role of carbonic anhydrase is rapid hydration of metabolic carbon dioxide formed in tissues, dehydration of bicarbonate ion in lungs, transfer and accumulation of hydrogen ions or bicarbonate ions in organs of secretion. Catalase and peroxidase. Both hydrogen peroxide and superoxide ion are reactive and toxic and may be produced in any biological system by auto-oxidation of many organic compounds in vivo. Catalase catalyzes decomposition of peroxide or superoxide ion to water and oxygen. This is one of the disproportionation reaction. Peroxidase catalyzes the oxidation of many substrate into peroxide and catalase helps in the decomposition of hydrogen peroxide into water and oxygen. Peroxidase and catalase have many similarities both in structure and mechanism. Both have high spin ferric heme groups surrounded with large protein molecules. Nitrate reductase. Nitrate is a source of nitrogen for most green plants and fungi. Nitrate is converted into ammonia, which is further utilized in synthesis of proteins, nucleic acid and other cell constituents. This process is known as assimilatory nitrate reduction. Nitrate may be used in place of oxygen as electron acceptor in anaerobic production of energy to certain bacteria. This is known as dissimilatory nitrate reduction. A flavomolybdoprotein, that is NADH nitrate reductase, and iron sulfur protein, that is ferredoxing nitrate reductase, converts nitrate into nitrite by first enzyme, and then nitrite is converted into ammonia by second enzyme respectively. Molybdenum is essential for reduction of nitrate to nitrite by assimilatory nitrate reductase from neurospora. Sulfite oxidase. The enzymatic catalyzed oxidation of sulfite occurs in mammals, plants and bacteria, and its role appeared to be in oxidative detoxification of sulfur compounds. The prosthetic group of enzyme is heme in most of the cases, and it has been shown that 2 hemes are present per mole out of sulfite oxidase, which consist of 2 subunits. During the oxidation process of sulfite converted into sulfate, one heme is reduced rapidly than the second, which also reduces much slowly. Studies show that it also contains molybdenum and the ratio of molybdenum to heme is 1:1. Xanthine oxidase. Its composition closely resembles to that of xanthine dehydrogenase, which is mostly found in chicken liver. The enzyme contains 2 molybdenum atom, 2 FAD molecule and 8 iron atom per molecule. Its role is that It helps in oxidizing xanthine into uric acid. It can also catalyze the oxidation of aldehyde to carboxylic acid. These conversions are shown in figure. These are the different reactions of xanthine oxidase. Like for example, hypoxanthine is converted into xanthine oxidase using water and oxygen molecules. After this xanthine oxidase can convert xanthine into uric acid, peroxide ions into p-oxynitrile and acetaldehyde to various acidic molecules. Aldehyde oxidase. This enzyme, also known as rabbit liver, have some similarity with xanthine oxidase. Both have identical content of molybdenum, iron and FAD, although aldehyde oxidase also contains two molecules of coenzyme Q. They have the ability to utilize molecular oxygen and generating peroxide anion. They have ability to catalyze the hydroxylation of aldehyde and N-heterocyclic substrate. Carp muscle albumins. Numerous low molecular weights albumins occurring in the white muscle of the fish are known to have biochemical properties such as in mediation of the effect of calcium in muscle contraction. Calcium 2 plus requirement for enzymatic activity is also well known. Staphylococcal nuclease is a single chain polypeptide enzyme of 149 amino acid residue which exhibit hydrolytic activity towards both deoxyribonucleic acid, that is DNA, and ribonucleic acid, that is RNA. Enzymatic activity of staphylococcal nuclease appears to be completely dependent upon calcium 2 plus ion when RNA or DNA is hydrolyzed. It has also been pointed out that higher coordination number usually associated with calcium ion and its ability to act as a bridge between anions makes it highly suitable for integration as structural component in biological system. In addition to these catalytic activity, calcium is a major constituent of bones and teeth. It is essential for muscle contraction, normal heart rhythm and nerve irritability. Metallothioneins, cysteine-rich proteins, have capabilities to bind metals such as mercury, calcium, zinc and copper. Those have certain affinity towards thiolate group. These are known as soft interactions. 7 metal ions can bind to one peptide chain and this makes metallothionein to work as detoxifying agent, especially against cadmium and mercury. In addition to this, metallothioneins also play important role in metabolic processes. Copper is the third most abundant metallic element in the human body after iron and zinc. In addition to electron transfer reaction in photosynthesis, it also helps to first oxidize galactose when present in galactose oxidase, second convert dopamine, that is adenols, to no spin furan when present in dopamine beta hydrolase. Urease from jack beans contain tightly bound nickel which activates the enzyme made up of polypeptide. The main function of urease is to catalyze the hydrolysis of urea to ammonia and carbonic acid. Many different bacteria are known to contain nickel which act as hydrogenase and catalyze the reaction, such as formation of water from molecular hydrogen and oxygen, production of methane, reduction of sulfate ion, etc. Glucose tolerance factor contains chromium 3 plus along with glycerine, cysteine, glutamic acid, nicotinic acid and is thought to be an essential trace element. Many of the bacterial enzymes such as formate dehydrogenase contain tungsten. Summary: nutrient metals from our diet are incorporated into blood. If blood levels are depleted, transported into cells if cellular levels are inadequate, or excreted if blood and cell levels are sufficient or overloaded. The sodium gradient hypothesis of glucose transport is linked to the sodium potassium ion transport known as sodium potassium pump. Lithium ion in form of lithium carbonate can be used in the treatment of manic depressive illness. Manganese is essential for normal bone structure, reproduction and the normal functioning of central nervous system. It is estimated that about 10% of human protein potentially binds zinc, in addition to hundreds which transport and traffic zinc. Copper is essential for maintaining the strength of the skin, blood vessels, epithelial and connective tissues throughout the body. About 99% of the body calcium is in the skeleton, where it is maintained as deposits of calcium phosphates in soft and fibrous matrix. Many more examples involving metal ions and their complexes playing important role in biological processes are discussed in this module.